All right, welcome to our explainer. Today, we are diving deep into one of the most fascinating and, let's be honest, one of the most important chapters for any biology student, the animal kingdom. Think of this as your complete masterclass, perfectly tuned for CBSE Class 11 and anyone with their sights set on NEET. We're going to break it all down, make it simple, and make it stick. So, let's kick things off with one big question that really drives this whole topic. Just think about it. You've got a simple sponge on one end of the spectrum, and then you have a complex human on the other. The differences are just staggering, right? So how on earth did this incredible diversity come to be? We're about to unpack the exact blueprint that biologists use to build the family tree of all animal life, giving you a really clear framework to understand it all.
So here's our game plan. First, we'll tackle the fundamental blueprint for life, the rules of classification. Then we'll map out the entire kingdom with a clear flowchart. After that, we'll take a journey through the non-chordates, then explore the dawn of the chordates, and we'll wrap it all up with a rapid-fire revision session completely focused on nailing your exams. Ready? Let's dive in.
Okay, Section 1. Before we can even begin to make sense of the huge variety in the animal kingdom, we need to understand the toolkit biologists use. Think of it like this. Before you can read a map, you need to understand what all the symbols and lines mean. These are the fundamental rules, the architectural principles of every animal body plan.
So we're going to be looking at 6 key features. First, how cells are organized. Second, the body's symmetry. Third, its embryonic germ layers. Fourth, something called the coelom, which is its internal body cavity. Fifth, segmentation. And finally, number 6, the presence or absence of a notochord. Let's start right at the beginning, with the most basic level, how cells get together.
What you really need to see here is this amazing story of increasing complexity. You start at the simplest, the cellular level, with animals like sponges. Here, the cells are basically just loose clumps, kind of like a simple colony where everyone has a slightly different job. Then, you take a step up to the tissue level, which you see in coelom traits. Now, cells doing the same job are grouped into tissues. Go another step up to animals like Platyhelminthes, and you see tissues grouping together to form organs, each with a very specific function. And finally, you get to the most complex level, the organ system level, where different organs work together in systems, like our digestive system. This is what we see from annelids all the way up to us humans.
Next up is symmetry. Think of radial symmetry like a pizza or a wheel. You can slice it through the center along any line and get 2 identical halves. This is a pretty good body plan for animals that stay in one place, because they can react to things coming from any direction. But then you have bilateral symmetry, which is what we have. There's only one way you can slice us to get a perfect mirror image left and right half. And this was a huge evolutionary leap. Having a distinct front and back, a head, means you can move with purpose, and you can concentrate all your sensory organs in one place. That's a massive advantage for hunting and just getting around.
Now let's talk about the embryonic blueprint. Some animals are diploblastic, like colinterists. They develop from just 2 main layers of cells, an outer ectoderm and an inner endoderm. But most of the more advanced animals are triploblastic. They have a 3rd layer, the mesoderm, that's sandwiched right in between the other 2. And this mesoderm, well, it's the real game changer in evolution. It's the source tissue for all the complex stuff. Muscles, bones, the circulatory system. It's what allows for much greater size, complexity, and movement.
And that brings us perfectly to the coelom. Now, the definition says it's the body cavity lined by mesoderm, but it's the type of cavity that really matters for classification. This leads to 3 main conditions. First, you've got your acoelomates, like flatworms. A means without, so no body cavity at all. Second, you have the pseudocoelomates, like roundworms. They have a cavity, but it's a false one because it isn't fully lined by that all-important mesoderm. And finally, you have the coelomates, that's us, with a true coelom. This true body cavity was a profound innovation. It gives organs space to grow, it cushions them from shock, and it lets them move around independently of the body wall. Super important.
All right, now that we have our complete toolkit of classification features, let's pull back and look at the big picture. This flowchart we're about to see is basically our map for the entire journey. It shows exactly how those features we just discussed are used to organize all animal life.
Look at this. See how it all flows logically? Right at the top, you have kingdom Animalia. The very first split, right here, is based on the level of organization. See? The cellular level takes you straight over here to phylum Porifera. Everyone else continues on. Then, the next major split is by symmetry, radial versus bilateral. And then if you follow the bilateral branch, the next split is all about the coelom. Animals with no coelom, a false coelom, and a true coelom. This flowchart is our roadmap. We're going to follow it branch by branch through the rest of our journey. And so our journey really begins. We're starting with the non-chordates, which is just this vast and incredibly diverse group of animals that, as the name suggests, don't have a notochord. We're going to follow the path of evolution, starting with the simplest forms and working our way up to the more complex.
First up, phylum Porifera, the sponges. These are about as primitive as multicellular animals get. Their absolute defining feature is this unique water transport or canal system. So imagine water pouring in through millions of tiny pores called ostia. It all flows into a central cavity, the sponge's seal, and then it exits through a single large opening called the osculum. This constant flow of water is everything to them. It's how they eat, breathe, and get rid of waste. For your exams, you've got to remember the examples: Cycon, Spongilla, which is the special freshwater one, and Euspongia, the classic bath sponge.
The next step up the complexity ladder is phylum Cnidaria. We're talking jellyfish, corals, and sea anemones. Their killer feature? The presence of cnidoblasts. These are specialized stinging cells that contain a little harpoon-like structure called a nematocyst, which they use for defense and to capture prey. They also have two main body forms: the polyp, which is usually attached to something and cylindrical, like a sea anemone, and the medusa, which is free-swimming and umbrella-shaped, like a typical jellyfish. Some species even alternate between these two forms in their life, a super fascinating process called metagenesis.
Now, closely related to them are the ctenophores, or cone jellies. So what makes them so unique? Well, two things, really. First, the way they move. They use 8 external rows of cilia, these cone plates, that beat together in shimmering waves. It's beautiful. And second, their amazing ability of bioluminescence. They can actually produce their own light. I mean, how cool is that? In the deep ocean, it's just an incredible sight. Remember the examples Pleurobrachia and Catenoplana.
Okay, we've now arrived at our first bilaterally symmetrical animals, phylum Platyhelminthes, the flatworms. Now, a lot of these guys are parasites, which brings us to a classic NCERT question. So let's hit pause for a second and think like an examiner. What special features would an animal need to survive as a parasite living inside another animal? The key adaptations here are hooks and suckers. These allow the worm to latch on tightly to the host's insides and stop itself from being, well, flushed out. And you can see two classic examples right here: the tapeworm, Taenia, and the liver fluke, Fasciola. You can really see that distinctive flattened shape. Now remember, these animals are triploblastic, but crucially they are acoelommates. They have no internal body cavity. And for excretion and maintaining their water balance, they have these specialized cells called flame cells, a really key feature of this phylum. The next big evolutionary step in body plans brings us to the Aschominthes, the roundworms. A huge leap forward here is the development of a complete alimentary canal, basically a digestive tube with a mouth at one end and an anus at the other. This tube-within-a-tube design is way more efficient. They are also pseudocoelommates. Another key point is that the sexes are separate. You have distinct males and females, and the term for that is dioecious. Many are pretty infamous parasites, like Ascaris, the common roundworm, Wuchereria, which causes the terrible disease elephantiasis, and Ancylostoma, the hookworm.
Next up, phylum Annelida, the segmented worms. There are two major advances that define this group. First, they're the first phylum we've met that has a true coelom. And second is true segmentation, or metamerism, where the body is divided into a series of repeating units or segments. Think about it. This allows for more specialized body parts and much more efficient movement using both longitudinal and circular muscles. For excretion, they use structures called nephridia. And of course, the classic examples are Ferritima, the earthworm, and Hirudinaria, the blood-sucking leech.
We have now arrived at the undisputed kings of the animal kingdom, at least in terms of sheer numbers and diversity, phylum Arthropoda. And that brings us to a direct question from NEET 2020. Pause and think for a moment. Which arthropod is known as a gregarious pest? The answer is Locusta, the locust. And gregarious just means they hang out in huge groups, in this case, massive crop-destroying swarms.
So, what's the secret sauce for their incredible success? Well, first, the name gives it away. Arthropoda literally means jointed appendages. These incredibly versatile limbs can be modified for walking, eating, sensing, you name it. They're also protected by a tough, waterproof but lightweight exoskeleton made of chitin. It's like their own personal suit of armor. And they've conquered every habitat with a whole range of respiratory organs, from gills to book lungs to tracheal systems. Important examples to know include the economically useful insects like Apis, the honeybee, and Bombyx, the silkworm, and don't forget Limulus, the king crab.
famously known as a living fossil.
Right, after the arthropods, the second largest phylum is Mollusca, the soft-bodied animals. Their body plan is really unique. It's unsegmented, and it's got three main parts: a head, a muscular foot for moving around, and a visceral hump that contains all the organs. Covering that hump is a soft layer called the mantle, which in a lot of species is what secretes the hard protective shell. For feeding, they have this unique organ called the radula. It's like a file or a rasp that they use to scrape up food. Key examples to remember are phyla, the apple snail, and of course the highly intelligent octopus.
Our final stop on the non-chordate tour is phylum Echinodermata, the spiny-skinned animals like starfish and sea urchins. Now this is where things get a little weird, in a cool evolutionary way. Their larvae are bilaterally symmetrical, just like us, but then the adults develop radial symmetry. But their most distinctive feature, the one you absolutely have to remember, is the water vascular system. This is an amazing network of hydraulic canals they use for moving around, grabbing food, and even breathing. Key examples are Asterias, the starfish, and Ophiura, the brittle star.
And with that, we've crossed a major dividing line in the animal kingdom. We are now moving into our own phylum, Chordata. This section is all about the key evolutionary innovations that set chordates apart from everything we've seen so far, and which ultimately paved the way for the rise of the vertebrates.
This table right here, this is pure gold for your exams. It perfectly summarizes the fundamental differences. The absolute #1 difference is the notochord, present in chordates, absent in non-chordates. Simple as that. Now look at the central nervous system. In us chordates, it's on our back, dorsal, and it's hollow and single. In non-chordates, it's on their belly, ventral, and it's solid and double. Chordates also have pharyngeal gill slits at some point in their life, and our heart is ventral, while in non-chordates, if they even have one, it's dorsal. This is a high-yield summary slide for sure.
And here are those defining features in the classic diagram. You can see the notochord, this flexible support rod running right along the back. And just above it, you see the dorsal hollow nerve cord. Here are the pharyngeal gill slits, and finally, the post-anal tail. The key thing to remember is that all chordates have all four of these characteristics at some stage in their life.
Now, phylum Chordata itself is split into three subphyla. In Urochordata, the notochord is only present in the tail of the larva. In Cephalochordata, it sticks around for their whole life. These two simpler groups are often just called the protochordates. And then there's Vertebrata. This is the group where that embryonic notochord gets replaced by a proper backbone, a vertebral column made of cartilage or bone. And this leads to a super important exam statement. All vertebrates are chordates, but all chordates are not vertebrates. Make sure you get that.
Let's start our tour of the vertebrates with the most primitive living class, Cyclostomata, the jawless vertebrates. These animals, like lampreys and hagfish, are ectoparasites, meaning they live on the outside of other fish, using their sucking, circular mouth. They don't have scales and they don't have paired fins. Their life cycle is really fascinating. They live out in the ocean, but they migrate to freshwater to spawn. After they lay their eggs, they die. The larvae then grow, transform, and head back to the ocean.
Now we move on to vertebrates that have jaws, starting with the cartilaginous fishes, class Chondrichthyus, think sharks and rays. They've got an endoskeleton made of cartilage and tough skin with these tooth-like placoid scales. A key feature is that their gill slits are separate and they aren't covered by a protective flap called an operculum. But critically, they lack an air bladder for buoyancy. Now think about that. It means if they stop swimming, they sink. No wonder sharks are always on the move. Examples to know are Scoliodon, the dogfish, and Trigon, the stingray, which, yes, has a poison sting.
We're now moving quickly from the water to the land with the superclass Tetrapoda. First up, you have the amphibians, which live a dual life and have a three-chambered heart. That brings us to the next class, Reptilia, and a direct question from NEET 2016. So pause and think, what's the deal with the heart in reptiles? The answer is a three-chambered heart is present in most reptiles, but, and this is a critical exception, in crocodiles, it is four-chambered. This is a huge deal because a four-chambered heart prevents the mixing of oxygenated and deoxygenated blood. It's a much more efficient system, and it's a hallmark of the warm-blooded animals that came next.
So, to quickly summarize the four major tetrapod classes, you have amphibia, like the frog rana, living that dual life. Then you have reptilia, the crawling animals like the cobra naja. Next up is aves, the birds, like Corvus the crow, defined by their feathers, flight, and being warm-blooded. And finally, mammalia, that's us, Homo sapiens, defined by having mammary glands to feed our young and hair on our bodies. And what unites all of them? They all have 2 pairs of limbs.
Wow. Okay, we have covered a huge amount of ground. So now let's do the most important part for your exam prep. A quick, high-speed revision to connect all the dots and really lock in those key differentiating facts.
Okay, let's do this. Rapid-fire review using this fantastic summary table. Porifera, asymmetrical, acolomate. Key feature is the canal system. Platyhelminthes, bilateral, acolomate, and defined by their flat body. Annelida, bilateral, colomate, and the big innovation, true segmentation. Arthropoda, bilateral, colomate, known for their exoskeleton and jointed legs. Echinodermata, radially symmetrical as adults, colomate with that unique water vascular system. And finally, Chordata, bilateral, colomate, and the defining presence of a notochord. Seriously, use this table as your ultimate revision tool.
And we are going to end on this one thought. I know this chapter can seem overwhelming with all the names and features, but the key is not just to memorize everything blindly. It's about seeing the patterns, that gradual increase in complexity, the key evolutionary innovations that define each group, and how an animal's body is built for its lifestyle. If you can understand the why behind the classification, the what will follow much more easily. That's all for this explainer. Good luck with your studies.